Welcome back, folks. It's time for a new chapter. Last time we talked about the structure and function of the membrane, and so now we're going to start looking at the movement of solutes across the membrane into or out of the cell. So we'll look at two things. We'll look at just plain old concentration gradients, and then we'll take a look at electrochemical gradients. Before that, let's just kind of review the problem that we're trying to overcome. The cell has this permeability barrier, and its internal components have this permeability barrier, the membrane. And the membrane, remember, is selectively permeable. It's got a hydrophobic core, and so small lipophilic solutes can move freely back and forth across that barrier into the cell or into and out of organelles. But larger, polar, charged solutes, they're not going to move freely. They're going to need to be facilitated in their movement across the membrane, so we'll come to that later. Now, this is a neat little slide that contains a lot of information. I don't necessarily think you need to memorize all of these types of movement and things that are going on here, but is what it does, I think, is it shows you the extent to which cells have to move things around in cells. It's easy just to think about the cell membrane and maybe a red blood cell with CO2 coming in and going out and oxygen coming in and going out. But a cell in a tissue is performing numerous functions that require it to move solutes into and out of the cell and into and out of compartments. And a lot of the time, those movements are interdependent. So let's just look down here at the chloroplast. The chloroplast here may be undergoing reactions that produce a molecule called glycolate, which has to be modified and metabolized in the peroxisome to produce 3-phosphoglycerate to go back into the chloroplast if we were dealing with a plant cell. So there are two organelles working together as a system, and so there has to be a lot of exchange of materials between compartments within the cell. Try not to limit your thinking just to movement into and out of the cell. So cells have to overcome this permeability barrier. Sometimes it's not a problem because of the nature of the solute being moved, and sometimes we have to facilitate diffusion using carriers and channels, and sometimes we actively have to pump things using active transport. So cells have to maintain a certain homeostasis, and part of that is maintaining concentrations of solutes which are compatible with their physiology and which are usually different from the external environment. And we can apply that argument also to the internal compartments. The concentration of certain molecules inside the mitochondria has to be different than the concentrations of the same solutes in the cytoplasm, and we can even think about how different the proton concentration is in the matrix mitochondria compared to the intermembrane space, and how the cell must maintain that membrane, that pH difference across the inner mitochondrial membrane. So all of this can be wrapped together in the word homeostasis. The cell is maintaining these disequilibriums of solutes inside the cell relative to outside the cell, and inside cellular compartments relative to outside the cellular compartments. But at the same time, there must be an exchange so that normal cellular behaviors can occur. So let's take a look at gradients to start with, and then we'll do something a bit more complicated with this towards the end of this chapter. Let's start with a simple lipophilic solute. So top diagram here, simple lipophilic solute will move down its concentration gradient, and so the direction of movement here is determined by which side of the membrane has the highest concentration of the solute, and then the solute over time will move down its gradient. So here the solute's movement is dictated only by the concentration gradient. So this is for an uncharged solute that can move across the membrane either by simple diffusion or by facilitated diffusion. Now here we're just showing simple diffusion, but this obviously applies to facilitated diffusion using a channel or a carrier. So if the solute can move down its gradient, that will be a release of free energy, so free energy can be released If, on the other hand, we have to move that solute back up its gradient, then that requires some work, and the delta G there would be positive. Now let's compare that with the movement of a charged solute, and that's what we're seeing at the bottom here in this bottom panel. So the movement of an ion is determined by its electrochemical potential, or its electrochemical gradient, and that is the combined effect of the concentration gradient and the charge gradient across the membrane. And so the active transport of ions across the membrane tends to create a charge gradient, and that's called the membrane potential, or Vm. And so the membrane potential will affect the a charged solute's movement across a membrane. So what's going on in the diagram here? Outside of the cell at the top, inside the cell at the bottom. So here is a false situation. It's a hypothetical. We've got a charged solute, an ion, high concentration of the ion out here, low concentration of the ion in here. There's no electrical gradient here. The solute will just move down its concentration gradient if it's allowed to. Now in this situation, we've got the positively charged solute outside. The outside of the membrane is positively charged. The inside is being shown as being negatively charged. So although there's a concentration force pulling the solute into the cell, there's also an electrical force pulling the solute into the cell. So those two forces combine, and movement here would be very rapid if the solute could move down through a channel, for example, or it could be carried into the cell. And so here the negative charge, the positive charge of the solute is pulled towards the negative interior, and there's a large concentration difference of the solute, and so the solute moves rapidly into the cell. Here we've got the flip of that situation. We've got a large concentration gradient, but this time we've got a membrane potential where it's negative outside, positive inside, and so the positive charge on the outside of the membrane in the form of the solute is being repelled from the inside, which is also positively charged. Even though there's a concentration gradient, this pulling force of the concentration gradient is being offset by the repulsive force generated by the opposite charge, or sorry, the same charge outside and inside the cell. So we can have the situation where the electrical force cancels out the concentration gradient force and there's no net movement. We can have the situation where the two forces combine and we get much more rapid diffusion of the solute when it's charged. So you have to start thinking not just about concentration gradients, but also about electrochemical gradients. This is super important if you take physiology and you're ever looking at neurons and things like that. So we'll wrap it up there. We're going to come back to electrochemical gradients when we look at the energetics of transport later in this chapter. So I'll leave it there, and I'll speak to you soon.